Since Monday, we heard some very interesting presentation about the effect of different management practice on soil organic matter. Specifically, two very interesting right before me on POM and MOM. However, there are still too few studies that evaluate the land use effect at broad schemes and national and regional schemes on soil organic matters. What I mean is, how can we know the effect of a management practice without evaluating at the broad schemes the land use effect? And how can we know the maximum potential achievable gain? Here the question we want to discuss is, what is the relationship between land use intensity and soil organic matter pools, including soil biological pools? Accordingly, the title of my presentation today is Investigating the Interaction Between Soil Organic Carbon, Soil Microbial Community, and Land Uses. And this might be redundant, but the objective of this presentation was to produce a broad-scale analysis that investigates the interaction between soil carbon stock, soil microbial communities, and land use intensity. The study area was Atlantic Canada, where 500 points were randomly selected and randomly sampled. However, in the presentation today, I'm presenting the results only for New Brunswick. Each sampling spot consisted of a 1 meter square area precisely assessed with a high accuracy GPS. This was made like that because we wish to resample the exact same spot every 10 years approximately to monitor climate change. And because we anticipate that at some point in the future we will be able to do maps at 1 meter resolutions. Each sample spot were separated into arthropods, nematodes, organic matter fraction, fungi, mycorrhiza, bacteria, enzymes, and soil carbon analysis, amongst others. Here we see how the samples were collected and dispatched. The sample for biological analysis were immediately frozen or refrigerated. Here in cat slides, we see the general analysis that were performed and the topographical parameter determined with satellite imageries. I won't go through that list because it's a little bit too long. In the studied region, the soil was shallow and rocky. Accordingly, biological analysis was made for the 0.15 centimeters, and the soil carbon stock was assessed at the 0.15 and 15.30. Below 30 centimeters, the amount of organic matter was negligible. The main soil interregions are potsoils, luvisols, leptosols, brunisol, regosols, and glysols. Each sampling spot was categorized as either wetland, mature forest that had been logged more than 30 years ago, young forest that has been logged between 15 and 30 years ago, forage pasture land use, forage crop rotation, crops without root crops, and crops with root crops, mainly potatoes. Here it's important to note that the wetlands were natural wetland, mainly under forested climate, and the forage pasture was originally under forest. We don't have natural pasture in Atlantic Canada. And all the sites across New Brunswick were forested until about 100 years ago. So the different categories are all originated for forest. It's also important to note that this is a survey, so the cultural relationships of the different categories need to be discussed with caution. The fractionated samples were sent to Beta Analytic and to Left-Bridge Research and Development for 14C carbon datings and Delta 13C relatively invert datings. Here in these two graphs we can see that our fractionated method was effective to divide different age group of the different organic matter fractions. And we can see that in the more labeled, the younger fraction, the POM fractions, there are still quite a few materials that is old in it. And we can assume that in the older fractions, the more stable fraction, there's likely some young material mixed with that too. In that first result slide, we see that the land use with larger soil disturbance contain less carbon on a percentage basis, and that it is mainly the label, the POM fraction, that is being lost with an increase in soil disturbance. But in the case of land use with agriculture, with the root crops, we see that we also start losing the more stable, the older carbons. Whereas the intermediate fraction of organic matter is relatively constant across all the different categories of soil disturbance. Soil organic matter carbon stocks is the difference between the input from plant residues to the output from microbial activities. When we analyze the results on a carbon stock basis, we see that the influence of soil disturbance or land use intensity is less marked than on a carbon basis. This is due in part because the land use with higher disturbance and higher land use intensity were compacted, which offset slightly the effect of a percentage decrease. So the decrease in carbon stock from the mature forest to the crop with root crops is about between 20 and 25%. And for each categories, as we increase soil disturbance, the decrease in soil carbon is between 2 and 5%. This indicates that the best management practice for a specific crop rotations could achieve a gain of between 2 and 5%. With the data collected, soil carbon maps with resolution of 20 meters were produced. Our students, Travis Prenell, made a poster with that I invite you to go check. It might look strange, but this is the first time that a high-resolution soil carbon map is made for Atlantic Canada. In that graph, or in that figure, we can see that when we zoom in, where the agricultural area are, we can see clear difference between the different management practice and land use on the soil carbon stocks. One of the cool analyses that was performed was the rock eval thermal stabilities, which is a pyrolysis that evaluates the overall stability of the organic matter. Here we see that in the crop land use with root crop, the proportion of stable carbon is larger than the other land use. And this relates to the earlier graphs where we were seeing this land use having a reduced amount of POMs in the first results that we see. One regression performed also highlight the relationship between POM and the overall carbon stabilities, that is a negative regression. Another regression showed that aluminium, which is abundant in our Atlantic pot soils, positively correlated with the thermal stabilities. This is likely due to OH groups that in the HL aluminium groups can strongly ligand with the organic matter at the clay surface, similar of what happened in the allophanes of endosols. The first biological parameters I'm presenting today is the soil microbiological total DNA yields. Here we see that in these ones, it decreases as we increase in soil disturbance or land use intensity. And interestingly, the DNA yields positively correlated with the intermediate fraction of soil organic matters, but not so much with the more labeled the POM and the more stable the mineral-associated organic matters. This might indicate that the more labeled and more stable organic matter fractions could be decomposed by more specialized, a reduced amount of organisms that are more specialized on those labeled and stable pools, whereas the intermediate pools, it's a generalistic organisms that is targeting on it. On each collected sample, soil mycorrhiza was determined. And here we can see that the forage pasture land use had significantly more mycorrhiza, endo and apsocular mycorrhiza than on the other land use. And if we compare the more intact and less disturbed forests compared to the crops had lower amount of mycorrhiza compared to the more intact and less disturbed land uses. When we see the results on the soil pathogens or general pathogens detected on the mycorrhiza collected samples, we see an increase in the amount of pathogens as we increase soil disturbance or land use intensities. This is likely due to the fact that the land use with more intensive agricultural practice had lower amount of plant, shorter rotations, which allows the soil-borne disease to accumulate year after year due to the short amount of plants in the rotations. For each sample collected, several enzymes was analyzed. And for most of them, we see a decrease in enzyme activities as the land use intensity or soil disturbance increase. And interestingly, the microtopographical features of the landscape at 1 meter scales had a significant influence on the enzyme activities. Similar to other parameters, soil macroanthropology tended to decrease with an increase in soil disturbance or land use intensities, except for one specific crops where there was an explosion of mites. But overall, the collembola in arthropods and others under the various arthropods decreased with an increase in soil disturbance. And overall, the collembola, the abundance of collembolas were at the maximums with a soil CN ratio between 15 and 30. But not everything had to easily and straightforwardly correlate with land use intensity or soil disturbance. This is the case of soil nematodes, where the relationship is more complex and it's not directly explained by soil disturbance. We have a PhD student, Erika Youngs, that describes and analyzes the different parameters that influence the distributions of the different groups of nematodes, which is predators, omnivores, herbivores, fungivores, and bacterivores. Soil respiration relationship with land use was also complex and not straightforwardly explained by the soil disturbance. Here, Jeremiah Valentin has a poster that details and explains the relationship between soil respiration, the 24, 48, and 3 days respirations. And in his poster, he explained all the different parameters that are controlling the soil respirations for each of the 500 sampling points. So coming back to our main questions. Well, we need intensive practice to feed our populations. But our results show that this is coming at a very high environmental impact for the soil, very high cost for the soil parameters evaluated. So accordingly, we have to be careful at selecting the fields that will be dedicated to intensive agriculture. And we have to be very careful at not wasting what is harvested in these intensive agriculture sites. And we have to avoid to do intensive overpopulation because we don't have much more new lands to expand into intensive agriculture on. So with that, I would like to conclude with the quote from a mentor of mine that once said, "Soil organic matter is like our bank account. We have to be sure to keep a healthy balance." And finally, I would like to say thanks to you for listening and thanks to all the students, many students that made that study possible.